DNA fingerprinting. To understand the process of DNA fingerprinting, or what is DNA fingerprinting, we must understand the following first. What is repetitive DNA? Repetitive DNA is a small stretch of DNA which is repeated many times in a chromosome. Now, what is satellite DNA? When these are isolated, used density gradient centrifugation technique, then repetitive DNA produces, or they show, high peaks, whereas the repetitive units which show low peaks are called satellite DNA. As you can see, the bulk DNA forms major peak and other small peaks are referred to as satellite DNA. So what is satellite DNA? During density gradient centrifugation, the small peaks formed which gets separated from bulk DNA producing high peaks are called satellite DNA. In simple words, satellite DNA is present in external, in the anterior end, or at the ends of chromosome, and they have repetitive units or repetitive sequences, and during density gradient centrifugation they show small peaks or low peaks. Now, what are the types? Basically of two types: microsatellite and mini satellite. Now, coming to the factors which help us to determine these two types. Those are composition of bases, means either AT rich or GC rich, then length of the fragment, and also, as you can see here, its number of repetitive units. Number of repetitive units. According to these, my satellite DNA can be of many types. Now, coming to polymorphism. What is polymorphism? In simple words, variation in genetic level, which results due to variation in genetic level of different individuals due to mutation which are not very harmful. Now, coming to the next part. First, I'll tell what is DNA fingerprinting. DNA fingerprinting is basically a technique which involves isolation of a specific sequence, of a few specific sequence which is unique in a specific person. Okay, let me simplify it. We know that in human beings there are 3 into 10 to the power 9 genes or base pairs present. In that 0.01% gene is unique for each and every individual. That is their phenotypic speciality. We can say that is genetic speciality. And this is basically done by or controlled by repetitive units or repetitive sequence, which we studied as repetitive DNA. So according to the sequence and number of repetitive DNA, each and every individual person can be identified. This is the method, or this is the this is the let me think the word. DNA fingerprinting is based on this. The whole thing I said till now, DNA fingerprinting is based on this, because in DNA fingerprinting, and as we know, DNA fingerprinting is basically used to determine criminal or to solve parental disputes. So DNA fingerprinting can tell us that from which specific individual that specific DNA is obtained, or even the relationship between blood relationship, that is parent and child. That relationship can be determined from DNA fingerprinting. Okay, like this. So what I said, it is these differences in sequence of DNA which make every individual unique in their phenotypic appearance. Now, polymorphism, variation at genetic level, these all we discussed. Now, what is DNA polymorphism? It is observation of inheritable mutation in high frequency in a population. I repeat, DNA polymorphism is observation of inheritable mutation in high frequency in a population. Now coming to next part, that is techniques involved. Basically two techniques are involved. One is VNTS. Here you can see VNTR, and also Southern blot. So variable number of tandem repeats is the full form of VNTR. It is a type of satellite DNA. So let me write techniques involved. Two. One is VNTRS, that is variable number of tandem repeats, and Southern blot. Now, name of the scientist who discovered or developed VNTR technique, that is Alex Jeffreys. This is the scientist. Name of the scientist is Alex Jeffreys. Okay. Now, what are the steps involved? Steps involved. So I hope DNA fingerprinting, what it is understandable. I repeat, DNA fingerprinting is a technique which is used in which repetitive DNA or repetitive DNA, satellite DNA or VNTRs are used to determine identity of a specific person. Now coming to the steps involved. As I said that what Alex Jeffreys did, he used satellite DNA as probe that shows very high degree of polymorphism. Polymorphism means variation in genetic level. Now what are the steps? First step, isolation of DNA. Second step, digestion of DNA by restriction endonucleases. Isolation first, the repetitive DNA is isolated. Actually the whole DNA or genome we can say is isolated, then it is digested or broken down using restriction endonucleases. Now separation of the fragments by gel electrophoresis or electrophoresis. Transferring, the process is called blotting. Transferring of separated DNA fragments to synthetic membranes, example nitrocellulose or nylon, those are the membranes. Hybridization using labeled VNTR probe. Hybridization is the process of forming hybrid, that is hydrogen bond in between two different DNA, means DNA coming from two different sources. Detection of hybridized DNA fragment by autoradiography, and then we will discuss this as it is shown in the figure. As I said that VNTRs are mini satellites, and it is a small DNA sequence arranged tandemly in many copy numbers. So what is VNTR? Steps involved done. VNTR is, full form we know now, variable number of tandem repeats is a small sequence of DNA repeated many times in genome. Okay. The numbers of repeats show very high degree of chromosome. Means this number varies from one individual to another individual. In one individual it can be 0.1, in another it ranges up to 20 kb. So the number, copy number varies from one individual to another, and the range is 0.1 to 20 kb. Now after hybridization with VNTR probe, the autoradiogram gives many bands of different sizes. These bands give a characteristic pattern for each and every individual, and it differs from individual to individual in a population except in case of monozygotic twins, that is identical twins. The sensitivity of the technique has been increased by use of PCR. PCR is a technique in which we just multiply or amplify a specific DNA sequence. Now see this: paternal chromosome, maternal chromosome, chromosome 7, both. These are the VNTRs, first shown in pink, then in yellow in chromosome 2, then in light sky color in chromosome 16. That is DNA from individual A, DNA from individual B, and these are the tandem repeats, as you can see, number. And now number of short tandem repeats which is obtained from crime scene, that is C. So which one it matches with, A or B? If you see, you can easily understand that C, this one matches with B, not with A, individual. So here also we can see that few representative chromosomes have been shown in this image, and understanding different colors are used, and it is clear that the banding pattern of DNA from crime scene matches with individual B, not with individual A. And DNA, obtaining DNA from a single cell is enough to perform this experiment, this analysis, that is DNA fingerprinting. And also application of forensic science also help us. Now at present many different probes are used to generate DNA fingerprints. Now one more point I want to add. So what is the use of DNA fingerprinting? It can be either used to determine identity of a criminal or to solve parental issues.